Good morning to all of you. And I think it is time that I will start 9:31. Also, I would just like to inform you that I'll start recording this from today. Hopefully, we can share this as well if somebody misses part of it or for posterity. All right. Yeah. And I will also share with you very soon a Google Drive where I think all the material will be also available so that you can resource from there. Yeah. Okay, maybe we begin today. Yeah, so I'll start presenting maybe a minute on any questions, queries, discussion. Anything that you would like to start off? Anybody? If not, maybe we'll begin. I'll share my presentation. We can go from there. So can you see the slide?
Yes, sir.
Okay, okay, good. So let's begin then. Yeah. What we started on Thursday is just the background, basic. And I thought I will just share with you the more or less the contours of this course, the way I'm going to cover microbiology basics, just the fundamentals, and we'll spend a little bit of time on taxonomy. Okay, so this is important for us. We'll work on taxonomy and then look at a few techniques, particularly useful techniques to control microbial growth. Then the actual growth of the organism, growth curve, growth rate, doubling time, etc. And in fact, this is place where it can become very, very quantitative in microbiology. Even microbial taxonomy can be very quantitative. We'll spend a little bit of time on that. Fermentation and scale-up. We'll try to go through this in a very straightforward manner so that you can go back and read from the books. But most of the time, I just want to make sure we spend time on the logic of it rather than spending time on just description of it. Okay, so I'll share the slide so that you don't have to waste time taking down notes. You can really participate in the discussion more frequently. Okay, so then the more important aspects of media and inoculum development, how people do this in an industrial scale, and then obviously the strain development and the biodiversity, which microbial biodiversity, which can be used for betterment of humankind. Okay, so these are the things that we'll spend. Of course, the last section will be very loaded. This is where we'll spend a lot of time on at a later stage, and maybe also a couple of seminars on this as we go along. Okay, so that's what we will do. Now, I thought I'll start with microbial taxonomy today. And as you can see, what is taxonomy in the first place? Okay, so you have a variety of organisms. Then obviously one way of trying to figure out, make it rational to understand is to group them together into the like kind of organisms in one group versus the more removed organisms which are not similar to each other into another group. So all this classification, taxonomy as we call it, it's very nice for, let's say, large-scale living beings like plants and animals. because we can see their form and structure, and visually we can distinguish. But bacteria, particularly microorganisms, it's not very easy. So that is where I think taxonomy has been a very important science, so to speak, because taxonomy of plants and animals is a lot more straightforward. Although even that was not very easy. Carl von Linné, when he started, okay, it was the beginning at that time. But then today, microbial systems to be classified is quite a bit of a challenge. Okay, so even today, so you can see that as we go along. So one of the prerequisites for taxonomy is that an organism should breed true. That means it should consistently show the same phenotype, same kind of features throughout its living state as well as after a number of generations. So the stability of the species is a very prerequisite for taxonomy. So you should be able to predict. Okay, so this kind of a feature means this organism. Okay, so for example, if there are two animals which look like a horse, one has stripes and one doesn't, then we know what the difference is, right? One is a zebra and probably other one is a horse, maybe. Okay, so we have some straightforward ideas as to what kind of superficial phenotypic bulk scale features of an organism because we observe them. Okay, but then based on this, we have two kinds of taxonomists who have two opposing purposes. A set of people called lumpers, who try to find out what is common to all these and then put them together into one group. So they are trying to highlight the similarities between groups of organisms. They are called lumpers. And then there are splitters who say even, so to speak, a single nucleotide difference, they would like to call them as two different individuals or two different organisms. But where do you draw the line? What is a genus? What is a species? Okay, so these are important features, and these are of course man-made. These are made by scientists. That this is where we call it a genus, we call it a species, and things like that. Okay, but the point is we have kinds of people who try to see commonality between different organisms and try to group them together. We also try to see people, scientists who try to tease it out and separate them further and further. So dissect out the individual features. So both are important in taxonomy, and obviously the earliest, great biologist was Carl von Linné. You can see 1700s. Okay, he is the one who introduced binomial nomenclature. This is important for us in microbiology as well, because each organism, each species is defined by a name and also has a binomial nomenclature for it. For example, genera and species, a genus and a species, to the typical example that all of you are familiar, I'm sure you're familiar, is E. coli, right? Bacterium E. coli. You're familiar with this? Guys are familiar with this, right?
Yes, sir.
Yeah,
Yes,
yeah.
sir.
So most of you are familiar with such names like Nisseria coli. So in fact, this name itself is due to the nomenclature that was proposed and eventually accepted. Okay, proposed by Carl von Linné. So this is called binomial nomenclature. First part of the name is the genus, genera, and the second part of the name is actually the species. So Escherichia coli is the name of this typical, bug from our intestines. And obviously, the person who discovered and first studied this bug is Theodor Escherichia. So obviously, the name has stuck that because it was Escherichia who discovered this bug and studied it, it's called Escherichia coli. Okay, so this is probably how things go. We're not going to spend too much time on this description, but I thought I'll let you know. Some of you might not know the background, so I thought I'll just discuss this. Okay, now many of these microbial systems, they are so valuable that, and there are so many of them, that just like you go to a library, you catalog the books so that it's easy access, right? So which book you want, you can easily pick up from there. So similarly, microbes, there are also a variety of them, and they're also classified. So there are places where these microbes are deposited for posterity. For example, reuse later, or for the sake of preserving them, so that it can be recalled whenever we want it. So these kinds of things are possible. So the collection of these organisms actually itself is a big business. It's a big business for a couple of reasons. First of all, you are cataloging all the species, all the biodiversity in the world at the microbial level. And then secondly, some of them are valuable. Obviously, there is money involved in it. Okay, so some of these industrially valuable strains are also preserved at some place. And whenever you today want to patent a particular process of a microbial process, you have to actually deposit the strain. And the organizations are also, recognized, so that these strains have to be placed in a particular accepted organization for the World Trade and WTO and international patenting purposes. So this is a very important business, okay, particularly for industry. Okay, and also when you go to a type collection center, okay, collection center from where you want to get an organism for whatever purposes, you actually are asked this question very clearly: are you going to use it for academic purpose, or are you going to use it for industrial purpose? Very clear questions are asked, because if you are going to use it for industrial purpose, then there are disclaimers that you cannot use this unless you give this royalty. You cannot use this for this purpose because it is already claimed. And also the cost of buying that strain is much higher if it is an industrial strain. For academic purposes, the costs are reasonably nominal, but you are also stuck by the clause. You have to sign an MOU that it is used only for academic purposes. So such collection centers, I'm sure many of you know, one of them is ATCC, well known, American Type Collection Center. A large number of bacteria are deposited here. All kinds of microbial systems are deposited here. But then there is also a fungal genetic stock center, which preserves and stocks all fungi. Also fungi whose derivatives, whose transformant forms versus mutants, a whole range of them. There is also a center in Europe called Euroscar, so which actually has a deposit of all the yeast strains, including every single deletion of every gene or double deletions of yeast. So you have a culture center from where you can just order a yeast culture with any gene deleted or any 2 genes deleted and things like that. Okay, so coming to the Indian system, okay, where at least we have 2 big centers where the strains are deposited. One is in NCL Pune, called NCM, National Center for Industrial Microorganisms, and then one in Chandigarh at IMTech, it's called MTCC, okay? Microbial Type Collection Center. So in each of these places, you will find a list and you find a catalog of organisms that are present there. So if we are doing research today, we are developing strains or we are isolating new strains for whatever purposes, okay, and if it is a new strain, then obviously it's nice and it's useful to deposit these strains in some of these depositories, so that for colleagues, for other scientists, and for posterity, it can be recovered from there, because maintaining a culture is not very easy. Yeah, I think I just closed the door. It's better. Okay, so the maintaining the cultures or for posterity, for future, is an important activity. Otherwise, you have to rediscover the wheel sometimes, so which is not a very good thing. Yeah, so the point is that as a part of taxonomy, you also have collection centers, you also have a proper way of storing material, okay, biological material for posterity, for the future. Okay, this is very important. Okay, now, of course, a large number of names of these organisms. You have to name an organism, because the same organism cannot be called, or you cannot call all of them as bacteria, right? E. coli is a bacterium, Bacillus is a bacterium, so various organisms. There is also other kinds of organisms which are fungi, or they can be protists, so a whole range of organisms. But then obviously you name them and you identify each organism. Just now I mentioned about E. coli, right? So this is the most well-studied experimental organism. The genome is fully deciphered. All the genes expressions are studied. So this is due to a study it's named after Theodore Escherichs, who studied this as E. coli. This is a bug for all the laboratory work. We do all our cloning in E. coli. Okay, expression of foreign genes, protein expression, things like that. We do in E. coli. So there are other names here. I'm not going to go into details, but I can share this slide with you. So obviously the name of the organism goes after the discoverer quite often, or in honor of that person. So there is a species called Pasturella, which is used in many industrial applications. It's due to Louis Pasteur. Okay, so Nisseria Gonorrhea, for example, it's a venereal disease for gonorrhea. It was isolated and studied by Albert Ludwig Neisser. Okay, so Salmonella typhimurium, your food poisoning bug. Okay, Daniel Elmer Salmon, who was the one who actually isolated and discovered this. So naming also tells you something. It tells you the origin. It tells you sometimes also, it allows you to give, a face to an organism in some sense. Okay, so Escherichia coli, Escherichia is the genus name, coli is the species name. So you have a variety of names here: genus, species, genus. This is the binomial nomenclature we talk about. Okay, for humans, we have Homo sapiens as a name, right? Homo means something, sapiens means something. Okay, so similarly, we have names for various organisms. Okay, whereas for bacteria, we have this kind of nomenclature. Okay, now that is important part for taxonomy, because you could have in Escherichia coli as an organism, but then it could have Escherichia something else, be another organism. For purposes like this, you will find that the same genus could harbor many species. Okay, so we'll come to those examples as we go along. Okay, there is one right here below. We'll just see. Okay, so I'm sure many of you know we have used. Some of you have heard of PCR. Some of you must have used PCR, polymerase chain reaction. An important component of the polymerase chain reaction is obviously DNA polymerase. Okay, and you must have come across the DNA polymerase tag, right? Tag polymerase. How many of you know of tag polymerase? Familiar?
Yes.
Yeah, people are familiar, right?
Yes, sir.
You guys know, what is tag polymerase? Okay, so now, how did the name tag come about? So this is another way of naming enzymes which come of these organisms. Tag is nothing but it's a DNA polymerase from Thermus aquaticus. It's a thermophilic organism, bacterium, and it's actually I will right now just mention that it is a archaebacteria, but we'll come to that later. But right now, just understand this bacterium is generally used as bacterium. The term is used bacterium is from Thermus aquaticus. Okay, tag polymerase. And I'm sure you will see that there are also enzymes people have used. Some of you have heard of PFU, which is an equivalent of tag but slightly better. It is obtained from Pyrococcus furiosus. It's also a thermophilic organism, and you can see that the first name, Pyrococcus, P comes from there. Okay, which is a genus, and furiosus, FU comes from there. So PFU. PFU is a DNA polymerase used again in PCR. Similarly, POV is another polymerase used also in PCR reactions from Pyrococcus woozi. So here are two organisms. Both are from Pyrococcus genus, but one is furiosus, one is woozi. Okay, so woozi actually name comes from ooze. Okay, Carl Woos, we'll come back to this later example. Okay, but the point is that you have enzymes coming from these organisms are also abbreviated like this. Okay, restriction enzymes, for example, I'm sure you're familiar, right? I'm talking about nomenclature, so you can just bear with me for another few minutes before we go to some more details. Okay, so BAMH1, how many of you have used BAMH1? Anybody use BAMH1, restriction enzyme? Very common enzyme. Have you done any lab on molecular biology so far? Anybody?
Yes, sir.
Yeah, so have you used a restriction enzyme?
Yes.
Which enzyme?
EcoR1.
EcoR1. So here is the one, okay? So BAMH1, EcoR1. Obviously, EcoR1, the way it is written, you can see that it has the hint of from which organism this enzyme has been isolated. Okay, so Escherichia coli organism R1, restriction enzyme 1. Okay, that's how the name comes about. And BAMH1 actually comes from Bacillus amylolyticus. Okay, so H1 is the number of that enzyme, but it is taken from Bacillus amylolyticus. So you can see that binomial nomenclature has percolated into many fields of biology, molecular biology, biotechnology, also in microbiology. Okay, microbial systems. synthetic biology. So you will get to see these quite often. You don't have to remember all of these names, but what I'm trying to tell you is just you should know the general logic of it. If you write something, you should know why it is written in a particular way. So when TAC is written, you know where it comes from. When BAMH1 is written, you know where it should come from. Okay, so these are the kinds of things. Or at least curiosity. If you use a new enzyme tomorrow, you should know where this enzyme is got from. Okay, this particular tool is got from. So you should know. Okay, so this is something which, as the title indicates, what's in the name. You should know how the names arise. All right. So when you do taxonomy, when you classify organisms based on their features, what are the characteristics that you use, okay, to classify them? Okay. First of all, microbial classification has a very, very long history, very, very long history, hundreds of years, not just last few years, But in fact, the earliest taxonomy of bacteria was done, and it was compiled in something called a Burgess Manual of Microbiology, bacteriology, systematic microbiology. And obviously it has evolved into many versions, and today probably more than 8th or 10th or 12th edition, and the 2015th edition is actually Burgess Manual of Systematics of Archaea and Bacteria. So I have shared this number with you. It has both email copy, online book, as well as a hard copy. So this is a resource from where you can look at all the organisms whose features are already characterized, studied, and what tools are used to characterize them, and then how, if you isolate a new organism, how can you place it into a particular category, a group, okay? So the classification of bacteria or microorganisms is done through this. But Burgess Manual is predominantly for bacteria, archaea and bacteria. But for fungi, this is not so useful. Okay, we'll look at that as we go along. But general characteristics that we use to classify microorganisms typically is morphology. Okay, but since they are all microorganisms, the morphology is not very easy to see because they're all microscopic. So for these organisms, we mostly use microscopy. Okay, so I'm sure in your high school or primary school, you must have studied cocci, bacilli, spirili, based on their shape, you can call bacteria into that. But that is one aspect of morphology that we're talking about, okay, which requires microscopy, okay? And obviously microscopy goes with additional features like if the organism is very transparent, quite often they are, so then you have to stain them and things like that. We'll come back to that. That is one level of sophistication further. So direct morphology through microscopy, then you take these organisms, grow on different media, like culture conditions, temperature, pH, light, etc. So then you will see that that also gives you a characteristic. So an organism grows at higher temperatures but does not grow at normal temperatures. Or an organism grows at normal temperatures but does not grow at high temperatures. So temperature itself can become a distinguishing feature, characteristic for this organism. And this is how you classify organisms based on, thermophiles, hyperthermophiles, mesophiles, psychrophiles. I'm sure you know what I'm saying, that thermophiles are ones which love higher temperature and they grow only at higher temperatures. Their growth rate is best at higher temperatures. So culture conditions also can classify. Okay. Now, I'll come back to this a little later, but let's go through this list. So you can also use chemical composition of these organisms as one way, and all these have been used in the Burgess Manual, by the way, from the beginning. Okay, so chemical composition like cell wall composition, fatty acid composition, you can just measure the fatty acid content, quality of the fatty acids, and then cell wall you can measure. So cell wall has crosslinks, for example, in bacteria, which are amino acids. Okay, so you have D-alanine, diaminopimelic acid, lysine, a whole range of amino acids, and those can also be used. Their composition can be used to define a particular organism's feature. Okay, and obviously the most well-studied example, classically, whenever you do microbiology, you do what is called as gram staining. You guys have done gram staining? How many of you know gram staining? Yeah, you have
Yes,
done.
sir.
So gram staining.
Yes.
So the point of it is that I'm going to tell you that I'm not going to spend time on telling you what gram stain is. Although this is an industrial microbiology course, microbiology is a background that you should know, gram staining you should know. Okay, so I'm not going to spend time. Basically, it is a way of differentially staining the cell wall of the organism, and accordingly you can classify organisms into two groups, which stain the wall, which are stained, which are not stained by gram stain. So you have gram positive, gram negative. Effectively, that staining tells you something about the cell wall composition. cell membrane composition. Okay, so that's the way one can study that. Of course, there are other kinds of stains as well. Acid part stain, fuchsin stain. So variety of stains are there. Then obviously you can classify organisms based on metabolism, production of acids, carbon dioxide, gases. Okay, they can produce a variety of acids like citric acid, acetic acid, whole range of acids. They can produce gases like carbon dioxide. Some of them produce, hydrogen sulfide and things like that. Some of them produce pigments. Some of them produce enzymes. So all these can be classified based on organisms can be classified based on this. Pathogenicity is another way of classifying, and I'll spend a little bit of time on this in a few minutes, which is based on an organism is pathogenic to a particular host. So its host specificity itself can be used to classify. Okay. So as some of you might know that we are right now in the pandemic, coronavirus, right? Coronavirus, of course, virology also is part of microbiology in many ways. Okay, coronavirus, attacks humans at this point of time. We know this variety. Okay, COVID-19. Some of you must have heard of H1N1, bird flu. How many of you heard of bird flu? Okay, it's also in the news today because I think in some places in Rajasthan, a lot of birds have died, and they think that the bird flu is back. Okay, so this virus, it's bird flu, but then at some point of time, this also crossed the barrier. It also started infecting humans. But that is an example just to tell you that a host specificity itself can be used to classify organisms. Okay, and obviously pathogenicity is well defined, and how you define a particular organism pathogenic, Koch's postulates for that. I'll just show you a slide just to brush up your background on Koch's postulates. Then obviously each organism would be antigenic, its surface features, cell surface features are different, and animals, particularly mammals like us, we generate a response, antibody response. We generate a host response. Antigenicity is used, and we do have serotypes. I don't know how many of you are treated for hepatitis, for example. Okay, it's a virus. Hepatitis is A, B, C. There are different kinds of infections, and each of these viruses you can have a vaccine against. That means you can have antibodies against each of these viruses. So we have interesting features like antigenicity that itself can be used for classification. Okay, so obviously reproduction itself can be used also to study, group organisms into different groups. In bacterial systems, most often it is asexual, binary division. But the moment you go to slightly higher organized life, higher, complex life, like fungi, you can classify based on the kinds of sexual cycle that they have. You can have phycomycetes, ascomycetes, basidiomycetes, and where there is no sexual cycle defined, you can put them into fungi imperfecta. So these are the kinds of reproductive classification also can be used. So lastly, of course, most important and the most recent is the genetic tools. They also can be used. We'll spend a lot more time on this at a little point later. Okay, so these are the list of characteristics. One or all of them, many of them are put together to identify an organism. Okay, is it a pathogen? If it is a pathogen, then we will use Koch's postulates and figure out which host it actually infects. For example, leprosy bacterium, Mycobacterium leprae. Okay, it attacks humans. Okay, but there are no culturable, hosts where we can actually culture and study this. Okay, it's very typical to humans, Mycobacterium leprae. Okay, whereas Mycobacterium tuberculosis, it can infect many other organisms, including humans. Okay, many other animals. Okay, so that is an issue that one can study. So we can study a number of these characteristics, put them together, and say, okay, these are the features means this organism. These are the features means this kind of a bacterium. Okay, so it should be. So E. coli will go through a number of these test tools, characteristics, and you can say, okay, this belongs to genus Nisseria, and it is most potentially E. coli bacterium. Okay, so that's how we can classify, characterize organisms. Okay, why is this important? It's important because tomorrow, let's say you isolate a new organism, you need to know, maybe it has a useful feature, but you need to know what kind of organism it is. For two reasons. One, this organism may be novel It may be not known at all before. So for that, in that sense, it is good science to do. Okay, but more importantly, suppose this organism belongs to a category of organisms. Okay, a group of organisms which we are already well known. Then your chances of using the same advantageous property in those group of organisms is very high. For example, see, if you have an organism which produces, let's say, antibiotics, okay, let's streptomyces, okay, streptomyces okay, as an organism, you know for sure that it is worth looking at other streptomyces because they may also produce antibiotics. So taxonomy is very valuable in guiding our research towards application. So that's why this is important. Although this looks very routine and mundane, but this has a great value. Most industries where they use microorganisms, they have well-trained, well-reputed taxonomists. Taxonomy is something which people don't like so much because they think it is routine and mundane, but taxonomists are very rare because of that, because you don't find any taxonomy so easily, but taxonomy is very, very valuable today. And you'll see for that reason today what people are doing, they're just going to genetic tools. Many of them don't even know how to do other things, okay? Although we are still teaching them in the laboratories, but I think well-trained taxonomists are very rare. Mostly genetic tools are used today, okay? I'll go on, show you more examples as we go along, okay? So this is a slide taken from a standard textbook. You can see that sources of taxonomic information and its value in different levels in taxonomic hierarchy. You have genotypic data, as I told you, in the previous slide. See genetic tools, so we'll put that on the top here. So chromosomal DNA, base composition, DNA hybridization, restriction enzyme patterns, okay, Rflp, AFLP, etc. And then whole genome sequencing, okay, DNA probes, DNA sequencing, PCR-based DNA fingerprinting, and then you have DNA, rRNA hybridization, nucleotide sequencing, ribotyping, and etc. Okay, so a whole range of them. We'll spend a little bit of time on this as we go along, but then you can see that at what level can you use these tools to go. You can genus or above, some of them are used. Species level, most of them are used. Subspecies, only few of them are useful. Okay, the broader the feature, less is it going to be dissect out into finer details, right? So you can see that if you see a bacterial cell wall, you can only say it is a bacterium or not, but you cannot say, because E. coli also has a cell wall, Bacillus also has a cell wall. But then you do gram staining, then you see the difference. Okay, so things like that. So this is genotypic data. Then you also have a lot of phenotypic data, which I discussed a little bit in the last slide. So you have proteins like amino acid sequence, electrophoretic pattern, enzyme, zymogram patterns, multi-locus enzyme electrophoresis, serological comparison, antibodies against these can be detected. Chemical markers like fatty acids, mycolic acids. For example, mycolic acids. You find mycolic acids mostly in mycobacteria. So you don't find an E. coli. So you can always say this is not an E. coli. This is not a E. coli. Okay, things like that. So lipids, polyamines, almost every member of the molecules, biomolecules can be used in some sense for classification. So you have proteins, chemical markers, whole genome chemical fingerprinting. Okay, you can use them. Whole genome fingerprinting, whole organism. The whole organism express features like morphology, we discussed, physiology, rapid enzyme tests. Okay, so most aerobic organisms, for example, have learned how, over evolution, have learned how to tolerate oxygen concentrations, because oxygen can be very toxic because of reactive oxygen species. So most aerobic organisms have superoxide dismutase, catalase, and other enzymes which can take care of reactive oxygen species. So very clearly, if an organism has these enzymes, it is an aerobic organism. Okay, if organism does not have this, chances are it is an anaerobic organism. So these are the very way you can classify organisms based on enzyme tests as well. Okay, oxygen requirements, physiology, and things like that. Okay, so I hope this is clear. Okay, so if assuming that this was a very interesting interview question. Somebody asked, how do you distinguish a horse from a zebra? So anybody has an idea how to distinguish? Suppose you are given a horse and a zebra. tell me, that they are different and they are different species. How do you figure out? Anybody knows? So this was an interview question. And the student quickly went ahead and said, I will take a small sample of the tissue, isolate the DNA. I will do restriction time Rflp or Rfpd. I don't know how many of you know these terms, but the person wanted to actually look at the DNA, go back to the genetic markers, genotypic data, one of these, and then say, look, this is a horse because we have a pattern for horse DNA, we have a pattern for zebra DNA. So they are two different genus. Okay. But what is missed in the process is one is not looking at the horse itself or the zebra itself. Just look at the stripes. The stripes are there, stripes are not there. That's a very important feature, right? You understand what I'm saying?
Yeah.
So
Yes.
you don't have to be doing everything by means of molecular or genetic markers. There are stages where you should go classical. Taxonomy requires that you should understand morphology. You should understand, one look at the organism should be able. So you don't have to always go and grind up the DNA and study its genetic makeup. Okay, so that's the reason why I'm spending this time to tell you that there are different bits of taxonomic information, not necessarily genetic, but others as well, which are very valuable in classifying organisms. Okay, making sure that these organisms work. See, for example, some of you might know that we think E. coli is a docile organism, but there are some strains of E. coli which are actually pathogenic. Okay, so if you just did the taxonomy in terms of just the DNA composition, Rflp, Rfpd, whatever or GC content or whatever, you will very quickly find that the two organisms are not very different. There are a certain set of small number of genes which will allow one to become pathogenic, other not to become pathogenic. Okay. So it's important to understand, have a good balance between the genotypic data as well as phenotypic data. So horse and zebra can be distinguished very simply by looking at the stripes of the organism itself, not necessarily go to the level of Rflp, is what I'm trying to tell you. Okay, so this is an important concept. You should understand all these things are used, okay, to get an idea about the kind of organism, its characteristics, and then you can put them into particular category of organisms or genus, class, or whatever. Okay, yeah. So the gross morphological features, as we discussed for microorganisms, is based on form. Okay, what kind of colonies they actually show up on a plate, on an agar plate, and if you look at a cross section on a plate, what kind of elevation you find. You can classify. You can classify these organisms based on the margins. What kind of margins that they have. I'm not going to go into details. I don't expect you to go, but just to tell you how classical taxonomy used to be done. Okay, so you could actually look at the morphology of the culture itself. You are not looking at an individual organism. You are looking at a colony. Okay, on a plate, and you can figure out. Some of these are very interesting features. Okay. Okay, so because taxonomy became very important, and many people were not aware of how this is done, there is also services available today where you can do taxonomy, not necessarily by the molecular tools, but also by phenotypic characterization. So here is an example. This is actually an advertisement taken from American Society of Microbiology News, 2000, November 2000. It's down here. What they have, this is a 96 well plate. Okay, and just to tell, and it's called Biolog. The company product is called Biolog. So what do they have is they have different media in small 96 well plates, and they inoculate the organism, and you will find what kind of organism is, each well will tell you whether it grows or does not grow. And if it grows, it gives you some color because of this is a vital color. Since the organism grows, it will change the color. Okay, some indicator. So you can see that typically you get a readout of this, and if you have mutants in some pathways, you can very quickly find out that, for example, this is not able to grow on arginine. this is able to grow on, this is able to grow. This requires arginine, this does not require arginine. This is the wild type, this is the mutant. Okay, things like that. You can actually get a phenotype read out based on whatever is your requirement. There are 96 conditions here. So you can have a large number of conditions, different sets of plates, and you can study. So this is a resource available. Okay, you can always look up, Google up, and find out some of these companies. So you don't have to do everything. No, you can just send the organism and make sure you can get this kind of data. So this is service, so to speak. Phenotypic microarrays you can get from Biolog. So another company called Microbial ID, which actually analytical services, which measures fatty acid, does fatty acid analysis of the organism, and it also does biological metabolic characterizations like Biolog, Crystal, MicroID, API system. It does comparative electrophoresis, ribotyping. All these things are done to classify the organisms. I hope you understand. Okay, so they will give you this set. It's almost like sending, your chemical sample to a place. Okay, get me an NMR data, get me a IR data and things like that. So you send your microorganisms, it'll give you a set of data, and you can sit and interpret that data to figure out which group it belongs to, why it's classical, reference to the earlier material, literature. Okay, so this is something which is done commercially today. Of course, if you do have, this is a very quick, 1 minute, let me just see. So we'll go back to that, okay? So these are different tools available for us today. There's something called Biolog, something called Microbial ID, something called Phenotypic Microarray from Biolog. So many of these are available, okay? Plus also you can see whether the organism is pathogenic or the organism is a disease-causing organism, the typical Koch's postulate. I'm not going to details because I'm sure you have studied this even in schools, okay? But just brush it up. So cause of a disease associated with the microorganism is called a pathogen, that microorganism is pathogen. And according to Koch's postulate, this pathogen can be isolate the microbe, grow it in a pure culture. And then pure culture is able to produce a disease in a fresh host, and you should be able to isolate the organism back from the infected host, and then it can do the cycle again. Okay, so this is exactly how Koch postulated that we can identify the pathogen. A large number of pathogens were actually identified like this. Anthrax was isolated by Koch like this, Robert Koch like this. Okay, Koch postulates anthrax, for example. So he says evidence required to establish etiological relationship between microorganism and disease. Microorganism must be observed in every case of the disease, must be isolated from that and grown in pure culture. So important to remember this, grown in pure culture. The pure culture then inoculated in the animal must reproduce the disease, and the organism must be recovered again from that. So it's almost like a cyclic process, which will confirm that you do have the cause and effect for the same organism giving you the same disease. Of course, look at isolating in pure culture. Now, this was done at the time by Robert Koch when organisms were actually grown in culture. Okay, even today we grow them in culture. But as time progresses, you start seeing exceptions, right? Koch postulates infectious agent at the time were bacteria, but we are not able to actually culture leprosy bacteria, Mycobacterium leprae in culture. It requires very complex media. In fact, it requires blood, for example. Okay, so for it to be able to grow. So prions, for example, which are protein infectious agents, obviously you cannot culture them. It has to go from organism to organism. So there are exceptions to the original Koch postulates, but I hope you understand what is the principle on which he proposed this kind of a test for a pathogenic organism identification. Okay, so obviously, although Koch's postulates are very valuable, what we have discovered a few things which are more interesting than what we actually thought as examples. Okay, so these again, pathology, pathology and pathogenicity is also one of the phenotypic tools. If you recall, okay, that is one of the phenotypic tools, right? Here, physiology, morphology, and as a part of that, okay, which characteristics is pathogenicity is an important tool that we looked at. But there are a number of genetic tools. In fact, this has become order of the day today for various reasons. We'll spend time, and of course, it's justifiably so, because it's also a very quick and more, I would say, deciphering tool because if you have a very sharp knife, you can cut into very small pieces, right? So genetic tools are much sharper at the molecular level than some of the morphological tools. But nevertheless, to distinguish between a horse and a zebra, you don't need a genetic tool actually. You know that one has stripes, one doesn't have stripes. But at the microbial level, genetic tools are very valuable. In fact, they have become very useful. I'll spend some time showing you this, how it works. All right. So what are the kinds of genetic tools that we talk about? Which two organisms, if they are different from each other, we will, based on genetic tools, we can put them into different categories, right? So one of them is DNA content, okay? The total amount of DNA per cell, okay? And the quality of the DNA, that means its base composition, GC content, for example. So the other gross genetic tool is TM of DNA, that is the melting temperature of DNA. And obviously, this has something to do with base composition. I'm sure many of you know this. Okay, so DNA hybridization or RNA hybridization can be done, okay, which will tell you about the sequence similarities in some sense, crudely. Okay, you can do Rflp and Rfpd. So anybody knows what is Rflp? Anyone knows what is Rflp?
Restriction fragment length polymorphism.
Yeah, restriction fragment length polymorphism. Basically, if you take an organism and you isolate its DNA, cut it with a restriction enzyme, okay, and you get bands on a gel when you run this on a gel, okay, and this banding pattern of fragments of DNA is unique because the restriction enzyme sites are unique on the DNA. Okay, if you have two organisms show the same pattern, we know that they are closely related to each other, or they are the same organism. But if the two organisms show a different kind of banding pattern, then obviously you know that they are not related to each other. So this is one rough way of trying to figure out how related two organisms are. And RAPD is a randomly amplified polymorphic DNA. Basically, restriction fragment length uses restriction enzyme digestion. Here, the variation is used as part of PCR. You take some universal oligos, universal primers, and do a PCR on the genomic DNA, and you get some fragments. Okay, these fragments are due to the localized annealing, localized hybridization of these oligos and small fragments of DNA amplified, which can be run. Again, these patterns can tell you whether the two organisms are related or not. In fact, RFLP and RFPD are very extensively used in paternity tests. I hope you know this. Okay, because the DNA coming from the mother and the DNA coming from the father have unique banding patterns. And of course, the child will inherit from both of them. So very quickly you can say, the paternity test can be very easily done. Who is the mother, who is the father? Testing the genetic background of an individual. Okay, this can be done. Okay, so then obviously ribotyping, which is one of the things which happened in late 70s and picked up very heavily. And in fact, this revolutionized the whole of taxonomy, microbial taxonomy. In fact, revolutionized the whole of taxonomy in biology itself. So we'll come to that. And the person responsible for that, Carl Woese, is a very famous biophysicist who contributed to taxonomy, particularly the ribotyping. What he did is basically he looked at the ribosomal RNA and ribosomal RNA DNA genes of the ribosomal RNA, and obviously he was able to sequence some of them partially, but today we can do full sequencing. So you see that bacteria, prokaryotes have 16S RNA, eukaryotes have 18S RNA, or rDNA as it's called, the corresponding DNA, and that sequence is unique, okay, for each organism, and that can be studied. All right. So this is ribotyping. So these are all genetic tools, and today most of the identification gets done by 16 or 18S rDNA sequencing. Okay, and the origin of that is due to Carl Woese, who looked at ribotyping in the early stages before sequencing. His work was just before sequencing. He was able to do sequence fragments, small fragments, but today we can do the entire sequencing of the genome. We can also do the entire sequence of the rDNA itself. Okay, so these are the genetic tools that we can really use today, and I'm sure you know each one of them, how they function. We don't need to go into details, do we? Can you read this up? You can find this in almost any textbook, okay? So we'll not spend time on that. But I'll just highlight a few things, if not all of them, a few things before we go to specifics, okay? I will spend a little bit of time on Carlboo's and Ribotyping, which is because it's important, it's contemporary. Okay, here is a slide showing you genome size and apparent organism complexity. An organism is more and more complicated. Obviously, it should have more information and it should have more DNA content. So this is an expected, anticipated thing. So here is a picture of the number of nucleotide pairs per haploid genome. Don't worry about diploid. Haploid we can compare because most bacteria are haploid, okay, whereas most higher organisms like mammals, okay, and reptiles, birds, most of the higher eukaryotes are all diploid. Okay, sexual reproduction all leads to diploid zygote. Okay, so you can see that the genome size, bacteria have the lowest size. Okay, and as complications, as things become more and more complicated, humans are here 10 to the 9. Okay, pair of nucleotides per haploid genome, 10 to the 9, 10 to the 10. Okay, in that range. But what you expect is that this value of DNA, amount of DNA, if you plot versus the complexity, you expect that it should be a straight line. It is not. In fact, it turns out there are outliers, a large number of outliers. That's called a C-value paradox. That means they have a lot more DNA, but they are not as complex, living beings as we anticipate them to be. They are not. For example, newt, an amphibian, has a lot of DNA. Okay, for example, lily, particular beans, okay, the plants have a lot of DNA, but they're not very different from Arabidopsis, which has this much of DNA. Okay, so there are minimum requirements for a particular organism's survival amount of DNA, but then there's also a lot of superfluous DNA, mostly non-coding DNA. Okay, so that's what is called a C-value paradox. So genome size alone is not enough to classify organisms, is one of the things that comes out of this. It is important, but not the only. Particularly when organisms harbor extra chromosomal elements, it becomes much more tricky. Okay. All right. So here is an example. Obviously, the amount of DNA will allow you to make a guess as to the estimated number of genes, estimated gene numbers in these organisms. For example, here most of them are eukaryotes. Okay, most of them are, so you can see humans, you can see C. elegans, you can see Drosophila melanogaster, you can see a plant Arabidopsis, you can see two fungi here. It turns out that if you look at the number of the genetic content, if you can see, the fungi also have, some of them have almost as much as human genome. But of course, not yeast, but some fungi have. But the point is the number of genes, okay, which are functional is what that matters in terms of complexity. Homo sapiens has a large number of genes. Arabidopsis has that, whereas yeast and fungi have much smaller. Plasmodium falciparum is a smaller number. So although this particular number is around 35,000, was what was identified when the human genome was sequenced in early 2000s. Okay, but today we know that this number of genes is not this large. It's probably around 22,000. Okay, but number of proteins could be large because of post-translational modification. So you will find the gene number estimate is another way of looking at complexity and classifying. All right. Of course, when you come to bacteria, also you will have variations like this. So here is another table showing genome size. As the genome size goes up, Homo sapiens is the largest in this list. Okay, protein coding, then number of chromosomes. Okay, so you can see that typically a bacterium has much smaller amount of DNA, smaller DNA, okay? and probably typically a single chromosome, whereas circular chromosome quite often. Okay? So this is also can be used in some sense as a way of classifying, but in fact this actually fails when you see things like C-value paradox. So the point of it is that you need to look at a variety of tools, variety of characteristics before you come to a conclusion. Okay, so that's what I'm trying to show you. So look at the bacteria themselves. Okay, so you have Pneumococcus, E. coli, Serratia, Mycobacterium fleae. Okay, so you see the GC content here. So what has been done here is GC content is plotted versus the melting point. So temperature, TM of the DNA. So as you can see that the more and more GC content, I'm sure you know that the GC content leads to tighter holding of the two strands. So more temperature is higher temperature required to denature the DNA. So the higher the GC content, the more is the melting temperature. So we can actually use this tool to find out the GC content. And it turns out that there are bacteria which have much higher AT content. There are the bacteria which have much higher GC content. For example, most streptomycetes have a very high GC content, almost 70 to 80%, 70% GC content. Okay, whereas E. coli is somewhere in between. Okay, so what does it tell you? If a bacterium has a higher GC content, that quickly puts you as one phenotypic characteristic into a group. But more importantly, at the molecular level, what does it tell you? The more the GC content means in terms of the genetic code, okay, it has a limitation on the third codon in the genetic code. That means most often it has to be G or C to maintain that GC content. Yes? So there is going to be a bias. But that also is a good way of classifying organisms. So there are organisms like Plasmodium, okay, falciparum, we have a very high AT content. So they are on the other side. Okay, so the wobble could always contain A or T predominantly in Plasmodium. But you cannot change the genetic code so much in the first two bases. The third can vary, right? So that is how you can classify. So this is an important feature. So this slide here, this part of the slide shows you something called DNA hybridization study and phylogenic relationship. So what has been done here is DNA hedgehog. Okay, hedgehog has been used, the DNA has been used as a standard, okay, and then its relatedness to human DNA, okay. So percent of relatedness to human DNA. So what you can do is you can take the DNA from these organisms, okay. You can see is many of them are mammals, most of them are mammals here. So you take the human DNA, hybridize, and you can see that hybridization can be possible. You can separate the double-stranded DNA and from the unhybridized DNA and find out the extent of hybridization. So percentage relatedness to human DNA depends on how much of it gets hybridized to human DNA. As you can see, you go closer to chimpanzees, the extent of hybridization is much higher. It turns out that chimpanzee DNA and human DNA is more than 99% similar, identical. Okay, so there are a lot of similarity. Okay, so the closer the two organisms are, the higher the level of relatedness, higher levels of through hybridization you can study this. Okay, so this is an important tool again, genetic tool, but a gross tool. We are not looking at the sequence; we are looking at the gross, similarities, right, at the level of the DNA. So this is another way of testing out the similarities. All right. So I think it's time. I'll stop here. Maybe take a couple of questions or doubts if you have. Then we'll continue in the next class. That's tomorrow. Okay. Okay. So have any questions so far? Anything at all want to discuss?
Sir,
Yeah.
Buddhadev here.
Yeah.
I have a question that in Burgess manual, what kind of classification is used? Is it a combination of everything or it is something particular?
So, good question. Burgess manual to begin with had a very few characteristics. They started in 1920s or whatever. Okay, very few characteristics. Today the Burgess manual is in 5 volumes. The 5th volume is all ribotyping, ribosome, RNA, rDNA, ribosomal, so 16S or 18S RNA. So it has different volumes and a whole range of tools are available. You understand that?
Yes, sir.
Okay, so Burgess manual is a compendium. You know what a compendium is? It was written by one individual, but eventually it has been edited and it has been expanded over time. Now it is a public domain available resource. So tomorrow you have a new tool to classify. It will also go into Burgess manual.
Thank you, sir.
Yeah. Okay. So anything else?
Sir, I have another question related to the course.
Uh-huh.
That is where we will find these slides and all those things.
Okay. So I have opened a Google Drive. once I am done with the class, I would put all this material there. Okay.
Okay,
So,
sir.
but more importantly, there are also references given to you guys. So you should go back and look at them. Some of these books are available even in open. Okay, you can look at them. Yeah.
Okay, sir.
See, this field is so vast, and I'm trying to cover microbiology before I cover industrial microbiology. So I'm going to go quite superficial on these things, but touch upon the most recent things. Okay, so that you get to know where to look for, what to look for. But if you have specific information, obviously you can Google up. Many new books are available. Journals are also available. Okay.
Yes, sir. Yes, sir.
Okay. Chala. Okay. So guys, see you tomorrow, maybe at 10:30. Yeah. Bye then.